Hello, good afternoon, dear colleagues. So let's resume the afternoon session. And just a reminder, we will continue here in this room with case studies for biologicals. And chemicals are in room 2D. So we will have this session until 3. And the session lead for this one on case studies on process validation and control strategy is Sean. So, Sean, please.
So we've heard a lot of information this morning already on process validation and control strategy. So now we're going to continue with a focus on biologics and some, really getting into the nitty gritty of some ways around the challenges we have and ahead of us. So the first presentation is going to be from Dr. Richard Keane, who's in Regulatory Affairs CMC at Biogen, and he's presenting on behalf of EBE, and is going to present an interesting idea in terms of the approach to setting specifications and tying in with what we were talking about this morning in terms of using the protocols and approaches like that to best effect and continued process verification. So thanks, Richard.
Thank you, and thank you for the opportunity to give this presentation. So the presentation I'm giving is about approaches to setting specification acceptance criteria, and this has been developed in conjunction with subject matter experts at Biogen, Amgen, and with UCB colleagues, and I'm presenting on behalf of the EBE Trade Association. So next slide, please. So the problem statement, as we saw it for the purposes of this case study, are that acceptance criteria for specifications, and we discussed some of this earlier, are often set strictly based on the range of values observed in clinical batches. And of course, products developed through accelerated routes are likely to have a limited number of batches at the time of approval. And sometimes what we've seen as an industry in the past is that acceptance criteria can be very rigidly set based on these very limited numbers of batches. They are not likely then to reflect the attribute acceptable range from quality, safety, or efficacy perspective, and also the future process capability. This is likely, and I think we've had discussions and examples of this already, to lead to failures of lots that are actually acceptable, potentially other aspects like shorter expiry periods and issues with continued global supply, because I think that's key to what we are trying to explore today. Next slide, please. So the case study, I'll skip over some bits and pieces, but effectively in the next 3 slides, we have an example of some data considering 2 key product quality attributes in the context primarily of a well-characterized biological product. These are high molecular weight impurities by SEC and purity by CESDS. As we've described, over time, as additional experience is gained with these products, manufacturing and analytical variability is expected to be observed. That's part of what happens in simple terms. But routine process monitoring applied via continuous process verification can lead to the identification of atypical batches and allow for investigation of root causes and indeed furthering the product knowledge. I'll have a little bit around stability as well, I know there's a stability session coming up a little bit later, but just to give some context to this as well. So I'll move to the next slide, please. So this first one, and this is just some data that we wanted to present that would kind of hopefully highlight some of the approaches that can be taken to data. So if you follow the continuum from the left-hand side of this to the right-hand side, this is the high molecular weight impurities acceptance criteria. And you will see at the beginning there the clinical lots, again, very limited clinical lots, moving into the PPQ lots, year 1, year 2, and year 3. So this, we debated back and forth. There's 50 data points on here in total. But what we can see here is that if we implement continuous process verification, and the process is well controlled for this attribute, if we could go in, if you were to set the specification limits based on the data seen in clinical, you would see a very small range. We focused here, because of high molecular weight impurities, on the upper limit, of course. Of course, there are considerations on the lower limit too, but we wouldn't go into that necessarily for the purposes of this discussion. But they're obviously important from trending perspectives as well as other perspectives. So what you can see here is that if we were to go in with, for example, a 3.5% specification limit at the time of initial submission, with the appropriate framework in place that I'll describe in a later slide, then we would be able to come back to the agency with a high degree of confidence in the future process capability and be able to present that. I think, in this example, you will see the green line is the mean within a particular area. The red line is kind of plus or minus 3 standard deviations, which is a statistical tool that is quite commonly used to view the data. And that specification limit can be brought down over time. So, and ultimately, based on current understanding and available literature, I think one of the other things we have to consider is that not all attributes are created equal, and I'll go into that a little bit later. But is there a true mechanistic difference or an impact on safety of quality, or safety or efficacy of something as small as a 1% difference in this context? And then there's also the consideration of prior knowledge, where we talk about things like higher molecular weight impurities is the terminology we've used, not just aggregates, because higher molecular weight impurities are not just higher order aggregates. They are likely primarily dimers, which often have been demonstrated consistently to be of less risk while often retaining biological activity. That would, of course, have to be demonstrated. So next slide, please. Very briefly on the stability front, I think what you can see here is the growing knowledge that you develop over the years of commercializing a product. On the left-hand side is a fairly typical idea of what you might get in an initial filing, with basically a clinical lot beyond sort of the 3-month time point, stability data available. Then over the kind of 3 years, which we focused on as a little bit of an arbitrary number, but I think it gives a good example. You build up your PPQ lots stability, you build up your annual lot stability. And in this case, for example, if you were to look at that first data set, you could quite easily draw a line through that and assume it's a linear relationship. But over time, you can see in this example that a biphasic trend for this attribute is demonstrated consistently with the longer-term trend of the clinical batches. And therefore, a specification limit on stability can be set with confidence that the trends will meet the specification throughout the shelf life. So next slide, please. Again, a slightly different example, but related to purity. So considering purity in the same way as we considered higher molecular weight impurities earlier, in this example, the final specification limit is tightened from sort of 93% up to potentially 94.5%. With confidence. What you can see here also is a data point that is atypical, and obviously that would be investigated. And in this case, as an example, we could say that no correlated manufacturing root cause is identified. And the batch conforms to all specifications, and you could, with some degree of confidence, potentially release that batch for commercial use. So move on to the next slide, please. So I think this slide, and I'll leave you to read this at your leisure, perhaps afterwards. But effectively, this talks to the point that each attribute needs to be considered separately and to its own merits. I think, as we discussed earlier, clearly immunogenicity is an important discussion when it comes to well-characterized biological products, as well as the methods that you employ to demonstrate the changes in attribute levels. So moving on to the next slide then, please. So the proposal here is that acceptance criteria for appropriate parameters and agreed parameters approved through accelerated routes should primarily be set upon the clinical phase specifications, documented and available risk assessments, as in available to the regulators and the assessors, which isn't, we know, always the case, prior knowledge that we discussed earlier also and the importance of that. And ultimately, the confidence in these acceptance criteria is enhanced by a framework that sits around this. This framework would include things like potentially post-approval commitments to reevaluate once sufficient knowledge is available, like the examples I gave earlier. Other things could be explored too. From the industry perspective, I think we'd be certainly very willing to provide things like higher level summary of trending data. Certainly, leveraging already available quality management principles such as APR, annual report processes. And the strong quality management system is fully implemented through continuous process verification. On top of that, there are other parts of the framework too that we've discussed today, such as control strategies, inspections, and even pharmacovigilance measures, which has been referred to, which could be an appropriate path for some of these attributes on occasion. Next slide, please. So to conclude, the ultimate goal of this proposal is to establish acceptance criteria appropriate to the level of available knowledge with a high degree of confidence that there will be no impact to safety and efficacy within the allowable range. We believe that this approach is feasible under the current network, but would require the context of establishment of acceptance criteria to be more broadly considered with these other attributes that we were discussing earlier, that framework that these decisions have to be taken within. The approach would clearly require closer interactions between the MAH and assessor and agency post-approval to ensure that continued monitoring is appropriate and that the quality expectations are set. And the ranges for these product quality attributes are generally well controlled for biotechnology products, well characterized ones at least, and the impact of these attributes on safety and efficacy is becoming better understood. And we discussed earlier some things like industry consortia looking at aggregates is an example that we could throw into that, for example. And ultimately, the acceptance of this approach would allow for confident, rapid development of these high-quality medicines and continued supply globally to meet the unmet medical needs. So my next slide is just an acknowledgement slide and for my colleagues who've helped to build this. And I think in the supplemental slides, I also have a link to a BPOG position paper, which talks to some of the principles that I've described here in this example. Thank you.
Many thanks, Richard. And next up, we have Dr. Teresa Pepper, who's Regulatory CMC Director at MSD Merck. She has over 13 years pharmaceutical industry experience in Merck, MSD, and Amgen, leading regulatory CMC teams with interest in vaccines, monoclonal antibodies, drug-device combination products. And she's also got 8 years previous experience as an assessor at the MHRA. So thank you.
So thanks, Sean, and thanks very much for the organising committee to invite me here to share our experience with the prime and breakthrough designations for the licensure of the MSD Ebola vaccine. We've been asked to focus really on some of the informal and formal interactions we've had with both US and EU regulators to accelerate our CMC approaches for product development and submission of the marketing authorization. So if you move to the next slide, I've just got some background on the project here. So we started out with the Public Health Agency of Canada's National Microbe Laboratory licensing a candidate Ebola vaccine designated V920 here to a small US biotechnology company. And we signed a collaborative agreement. They signed a collaborative agreement in 2014 with Merck MSD, who took on the efforts of research, development, and manufacturing. The vaccine itself consists of a vesicular stomatitis virus, which is an animal virus which causes flu-like illnesses in humans. And it's been genetically engineered to contain a glycoprotein from the Zaire Ebola virus so that the genetically modified live virus vaccine can elicit an immune response to the Ebola virus. We were granted a prime and breakthrough therapy designation in June 2016. And whilst our experimental vaccine has undergone numerous clinical trials in Africa, US, and Europe, and was used in the latest Democratic Republic of Congo outbreaks this year, which in itself caused 11,000 deaths, it's still to be licensed. So we're planning to license, to submit the marketing application to the MEA and FDA during 2019. Then we will seek pre-qualification by the WHO and licensure in African countries where the Ebola is endemic. So if you move to the next slide, really, this is the situation we were facing. There was a clinical development program was conducted very rapidly. We had 12 phase 1, 2, and 3 trials ongoing during the 2014 to 16 Ebola outbreak. We had the results from an efficacy and safety ring vaccination from 2015 in Guinea, which showed the vaccine to be highly effective, and because of the ethics of the situation, we were not able to use placebo-controlled trials, but we used a ring vaccination strategy where you vaccinate a ring of contacts around an individual Ebola case, and then each of those contacts is in turn vaccinated as well. And so we were able to show efficacy, and WHO recommended the vaccine for compassionate use. And to date, we have more than 27,000 eligible and consenting people being vaccinated. The clinical lots that were made at a contract manufacturer, and clinical consistency was performed using those lots before scale-up of the process and transfer to the final manufacturing facility. In the next slide, you can see some of the CMC approaches to accelerate product development. So since MSD began its work in 2015, our goal was to build capacity to manufacture the vaccine using these expedited approaches, including the use of the clinical formulation to avoid development delays. This presentation actually focuses on our analytical comparability bridging strategy to support the process scale-up from the contract manufacturer to a pilot plant and then transfer to the final manufacturing process to a dedicated EU manufacturing facility. And it also talks about our tailored validation package, which is some of the things that Sean has talked about already. Throughout the whole development, we shared advanced CMC data with both the US and the EU agencies to gain access to their timely regulatory advice that we could act upon immediately ahead of the MAA submission. The next slide outlines a two-step comparability approach that we used. Step one was a retrospective, informal assessment of comparability between the clinical lots used in the ring efficacy trial manufactured at the contract manufacturer when MSD became the sponsor, to Merck's pilot plant facility, which was used to manufacture emergency lots to support ongoing Ebola outbreaks and clinical trials. The contract manufacturing process was transferred to the pilot plant with minor process and equipment changes only for facility fit, and it was scaled up to the commercial scale there. And then clinical lots from that pilot plant were used to evaluate clinical consistency, as the contract manufacturer at that point was no longer manufacturing the vaccine. The step two required a prospective comparability protocol that we typically see. to bridge between the clinical batches used in the efficacy trial, the ring vaccination trial, from the contract manufacturer, and the proposed commercial site in Europe. The protocol we had reviewed early by the FDA and the EU regulators ahead of its implementation. And this early dialogue with those regulators accelerated generating the comparability data that we needed to support the global marketing application. In the next slide talks about the parallel validation activities and our tailored approach. So typically, traditional process validation for a biological active substance and its resulting drug product requires a minimum number of batches, and that's typically 3 consecutive full-scale drug product and 3 drug substance and drug product PPQ lots to be submitted in the MAA. And although we had such a high unmet medical need for this vaccine, I mean, the outbreaks are continuing. 1 in every 2 people who are infected with Ebola will die. The idea of concurrent validation or continuous process verification was very challenging for this vaccine, given its complexity and its uniqueness, as it's a genetically modified live virus vaccine with shortened accelerated timelines. So instead, what was agreed by the US and EU agencies was a tailored expedited validation, whereby we had ongoing in parallel overlapping drug substance and drug product validation activities. And by that, what I mean is that as the drug substance PPQ lot 1 data were available, those data were used to start the drug product PPQ lot 1, such that there was overlapping drug substance and drug product PPQ lots being manufactured. As you see there, the amount of process validation data and the timing of the submission is going to be determined after discussion with both agencies, but thus far what we have agreed is the FDA has requested the drug substance PPQ and drug substance comparability data to start the review clock. The timing of the submission of each of the drug substance and drug product PPQ dataset, either within the initial MAA, during review or post-approval, is still under negotiation, but it does appear that both agencies are amenable with regards to what PPQ data are required to support approval and whether some DP PPQ data can be deferred to post-approval. And to support that approach, the FDA GMP inspection timing will coincide with drug substance and drug product PPQ manufacturing taking place at the commercial site. In the next slide, you can see the clinically relevant control strategy. We had limited batches from the contract manufacturer, which were used in the ring vaccination trials. Potency specification were derived from clinical lots with the lowest potency to ensure efficacy and with the highest potency to ensure safety, but it was using a developmental potency assay. So this figure really shows the commercial potency release limits are derived from testing the vaccine retained samples with the commercial assay to establish the upper and lower limits. And whilst doing that, we had to consider how lots were actually being used in the field, including the administered dilutions, stability losses due to time out of the minus 70-degree storage condition, given that the vaccine is intended to be used in tropical countries, in isolated places, and the challenges of getting the vaccine to patients. So finally, just to move on to the interactions with agencies, in this next slide, we have throughout the whole licensure under both accelerated schemes, we have had frequent formal and informal interactions with both EU and US regulators from the initial onset of the prime and breakthrough therapy designation in June 26 to several years, which is several years ahead of our marketing application right through to date. And the right balance, obviously, to achieve is to get feedback on key obstacles and risks. So it's very useful to have the flexibility of both informal and formal interactions with both agencies to ensure the company is aligned. The informal meetings, of course, were more flexible and really very valuable. They weren't restricted by any formal meetings and timelines. The key issues were discussed in a timely way. And if you compare that to the standard centralized scientific advice taking maybe 70 days or even 40 days in the truncated process, formal briefing packages, presentations, this is a very good approach. What we did was we provided advanced CMC data and questions to the FDA under the IND amendment scheme that was mentioned earlier, and equivalent submissions were made to the EU regulators via UDRALink, and we always provided a formal RTQ document to capture answers to regulators' questions. Both FDA and EU responses were very timely. For example, FDA always responded within a month or even a week when we had multiple reviewers looking at the package. And specifically in this case, MSD received feedback on analytical method validation for the key assays. the adventitious agent control strategy given the uniqueness of the vaccine, and the risk assessment for proposed control strategy. Most interestingly was for one key CMC topic that we identified late in the process that could have impacted approval. We were able to provide a draft Module 3 section for early review to the US and EU reviewers, and this section was then further reviewed by the BWP. So with timely feedback on this critical issue in particular, we ensured global alignment without risking changes to the strategy close to submission of the MAA that would be hard to accommodate at that stage. Not only were the regulators willing to meet with us, but they also discussed these key issues multiple times to ensure we had full alignment on all our commercial plans. And by having the early prime and breakthrough therapy engagement for this critical topic, the regulators specifically assisted MSD with the minimum filing Module 3 registration details for this topic. This was very helpful. And then finally, in summary on the last slide, what we found then is that frequent informal and formal discussions with the EMA and FDA under prime and breakthrough therapy have facilitated development of an Ebola vaccine. The transparent and open communications we had between the company and both agencies, and particularly the review of the advanced CMC data and the rolling submissions with the FDA, they allowed the continued progress of this project and limited the potential risk to submission of the marketing authorization by letting us align and understand what the regulators were expecting. In particular, the feedback on the key aspects of the control strategy, the comparability protocol, including setting of acceptance criteria and the clinically relevant specifications through these expedited and often informal agency reactions enabled our company to focus on really what were the remaining items that needed to be addressed in the dossier. And specifically the comparability protocols that we discussed under the IND by the CBER and FDA and in the EU under PRIME was really valuable in getting us to the commercial site ahead of marketing. Thank you very much. And in the last slide, just thank you to my colleagues, Kim Hassis, Janthi Wall, Scott Willens, Christy Moore and Catherine Sledgers for help with the presentation.
Many thanks for that, Teresa. So we'll have our panel discussion now and just introduce the other panellists. So from FDA again, pleased to welcome back Manuel Lucana and Laurie Graham. In addition, Dr. Andrew Burns, who from FDA, who's Chief of the Gene Transfer and Immunogenicity Branch in CBER, in the Office of Tissues and Advanced Therapies, and also Andrew Leonard from Amgen, from their EU Regulatory Affairs for CMC, representing EPVIA, TGEG, and the EB Biomanufacturing Working Group. So we'll open the floor to questions. In particular, to hear some of the solutions in terms of the challenges that we face, particularly with setting specifications and control strategy, and how we can really get past this when we have only limited number of batches. Maybe I can start if there's no burning questions, but I'd be interested to hear. So, Richard, I liked your concept of how you would link the CPV or the ongoing process verification over time, you know, where you nicely showed what the true variability of the process might be, which isn't reflective in maybe the 3 PPQ batches or clinical batches. 2 questions in terms of, in the file, how would you plan to initially justify that wide specification? How would you come up with, you know, the wider specification that you will ultimately narrow? And you know, for the assessor, when they're writing the report, they go, okay, we have this specification. I'm happy to accept this for X reason, but really that reason would have to be kind of hopefully justified by the applicant. And also, how do you see the protocol being put into the dossier in terms of what's the best way to do that, in terms of this is stuff that normally happens in the background in terms of your monitoring batch data over the years, and how do you see that fitting into the dossier?
Yep, thank you. It's clearly a challenge. I think we've started to address some of these topics a little bit earlier. I mean, in terms of what we would actually present, I think what I'd like to maybe open with is we'd like to speak to the regulators more about what you would need to see and at what level of detail, because I think, you know, we could provide, you know, a full breakdown of the quality management system, our SOPs for investigations, OSs, all those types of things. But would we need to do that? And I think that's the challenge. And I think, you know, it's bringing together all of the stakeholders within the framework. So we've got the inspectorate group, we have we have others, and I think we need to try and leverage that appropriately to be able to give these context. So it's not just a numbers game. I think that's the point I'd like to really try and bring across with this. It has to be seen within the broader framework. Now, quite how we could demonstrate that is going to be a challenge. I mean, as we discussed earlier, we can't just provide hundreds and hundreds of pages because that's not meaningful. And I think, you know, it's going to be in my mind, it's going to be a collaboration and a learning process as we start to try and implement some of these types of approaches. I mean, in terms of the specification, I think in the proposal I've said, you know, we do need to present that in a meaningful way. I do take your point absolutely, but I think it would be primarily based on your clinical phase specification because I think it has to be, because I think that's essentially all you have at the point of approval. But with a proposal built into your dossier, be that a plan, be that a protocol, be that a post-approval change management protocol, that says that we will confirm that once we have the appropriate amount of data. Now, how we confirm the clinical meaningfulness of that data, because I can see Matt's pointing at me already, is something that is obviously a challenge, and I think it's going to be difficult. But I think we can find ways of addressing a lot of these topics. But I think we're at the start of that process, and I think it has to be a collaboration to move it forward.
Matt, did you want to?
No, just as you said, that because when basing it on the clinical trial specifications, that yes, there you have an argument to saying this may be acceptable, and you are going to actively look for that in your clinical studies, and here you don't have the possibility if you have it. So I agree. This is what will be, but again, it's. I think when we are looking at the figures we are talking on fractions of percentages sometimes. Then, okay, maybe this is not. If you are saying that you have 80% and your clinical data was 95, we would have been difficult. If you want 94, perhaps that is more doable. And again, taking into it, this may be a little bit different kind risk-benefit in the sense we have an unmet clinical need.
Just on the specifications, that idea that you would approve on clinical specifications, you may have clinical spec of 50, say potency 50 to 150% specification, but all your batches might be 98%. Mm-hmm. So even though it was approved, the specifications were approved, you never got batches that were anywhere near the very liberal specifications that we might allow in clinical trials. So there's a certain disconnect there.
So I mean, the way I sort of look at it is, the clinical specifications aren't just numbers that are plucked out of air. They have to be justified in some means, and that we would largely be using our prior knowledge and product knowledge for that justification. So the gap is the clinical qualification, and we would hope that that could be appropriately mitigated by the commitments that Richard had mentioned. And that's similar to the sort of commitments that we have in stability for clinical trials in trending and reporting out the specs, investigations, and so on. So also embedded in there is a description of the quality management system.
I think it's
and
really
that sort of totality we would hope to be sufficient for the interim until we do have that clinical experience and data to fill in the gap.
Yeah, I think it's key bringing all that information in. As if you just say in the dossier, these are the specs we had in the clinic, so that's it. You're going to get some questions on that. You need to bring in the prior knowledge and the other sources of information to justify why those clinical specifications are still relevant for a commercial.
So on the point of clinical trial specifications, it's not uncommon for ATMPs, which maybe the clinical trial has only 12 patients, and very rapid timelines. Assays are introduced relatively late in clinical development, like a potency assay or a new assay. Often we see specifications of report only, so it's not particularly helpful in that case. But on the point of how much justification to provide, my bias is always to include more rather than less, because we can always skip over things that we don't actually need. But if it's something that we need that's missing, at least to delays, we have to request that information or look at it during the inspection. It's always helpful to have more rather than less, in my opinion.
Jean-Pierre?
Yes. Regarding the quality management system, it's part of the inspection, so it's clear that during the inspection, the inspectors will assess the entire pharmaceutical quality system. So I think that by using the interaction between assessor and inspectors, there is an exchange of knowledge about this. So in order also to avoid that 100 pages should be provided and also to assess by both, so assessor and inspectors, by challenging each other, it's the best way to move forward.
Yeah, I guess you're bringing in aspects of ICHQ 12, which we won't discuss here, but I mean, you're bringing in that aspect of more the assessor and the inspector being involved in looking at the PQS, because you cannot write a PQS in a dossier. It's you have to be on site to really have a proper understanding of it. But you can bring in elements of framing how the PQS will be used in practice to get the data.
So I'll just say from a CDER perspective, one of the foundational principles of OPQ was sort of integration of inspection and application. So we now have our OPF facility and process reviewers that are on the team, and we also have our field investigators, ORA, that has now joined the application team. So everybody is part of the conversation about what's going on with the application.
To add to what Lori just said, for biological product, there is always a product reviewer that accompanies somebody from either from OPF or from the field investigator in the pre-license inspection, and it's usually the person that is most expert with the application.
Any additional questions? Or are we finishing on time then? So, amazing. So many thanks to everyone for a good contribution.
Sorry, just, just wanted to have a comment or a question about the Ebola presentation. So how do you feel it's really representative of an accelerated access, considering the real emergency situation? So is it really the interaction, the content, is it really representative of an early access? Or was there any specific, let's say, additional interest because of the emergency?
I think it's probably it's maybe an outlier in terms of, the extreme unmet clinical need in this case, and the real desire to have the product on the market means that, all the stops were pulled out in terms of, rolling review and so on. I don't think practically that would be possible for every product, considering the workload involved probably even on both sides. So it's good to have, what you could strive for in the cases of the real unmet clinical need. And there are certainly aspects in terms of, we keep talking about communication and more interaction. These are the positive things that come out of that. Whether that level of interaction is probably not really possible for every product, but I think it's on a spectrum, a continuum, depending on the benefit risk and the unmet patient need.